This is Ameya Bhope from the Department of Electrical and Computer Engineering at McGill University, and I am pleased to present our research, Network-Guided Gene Signature Analysis using the Generalized Ranking Distance method, developed under the supervision of Dr. Ameya. Gene expression signature is commonly used to provide a picture of the gene expression programs of cells in specific conditions, such as a tissue type, a disease, or in response to a stimulus. Gene signature analysis allows us to map gene signatures of a condition to signatures of other conditions. For example, by finding the best positively matching signature, we can annotate cells in single-cell transcriptomic studies. Or by finding the drug signature that inversely matches a disease signature, we may be able to identify drugs that can reverse the gene expression patterns of the disease. Current approaches for gene signature analysis can be categorized into correlation-based and machine learning-based methods. However, one major drawback of both these categories is that they fail to capture functional and biochemical interactions of the genes in a cell. To overcome this shortcoming, we developed a method called Network-Guided Gene Signature Matching, or NGSM, that can take into account three types of factors while doing gene signature analysis. The first factor is pairwise gene relationships. For example, in the figure, if G1 and G2 are closely related to each other by maybe belonging to the same pathway, the mismatch between the two signatures might be biologically less important than in the case where they are unrelated. The second factor is element-wise importance. In some cases, one might be interested in the position of some specific genes, and swaps in the position of those genes must be emphasized. The third factor is positional importance. One might be interested in the top or bottom of the list, for example, corresponding to the upregulated and downregulated genes, and changes in the genes in these positions need to be emphasized. Our NGSM method enables incorporating all three factors. The NGSM method takes 5 inputs: a query signature, a signature catalog containing many signatures, a list of biological pathways or gene level networks from which a pairwise distance matrix is obtained, gene positional and gene specific weight vectors for the query signatures. The first step is to obtain a pairwise distance matrix. In the case of pathways, for example, reactome pathways, the distance between two distinct genes is 0.1 when they belong to the same pathway and 1 if they don't. For biological networks, such as string experimental protein-protein interaction, each node, here each gene, is embedded using a graph embedding technique such as large-scale information network embedding or LINE. From the gene embeddings, a distance matrix is obtained by calculating the cosine distance between the embeddings. Next, we use the generalized ranking distance with Spearman-Foot rule to include all three types of weights to compare the query signature to each signature in the catalog. The method calculates scores for each comparison, which are then ranked to obtain the most similar and most distinct signatures at the ends of the list. For the generalized ranking distance calculation, 4 factors need to be considered. Starting with the rank list, the position of each element in the rank list and its corresponding importance weight of each element, displacement of an element in the rank list, and gene-gene distance values obtained from the pairwise distance matrix. Consider 2 lists to be compared: the query signature and catalog signature, consisting of 5 elements each. Since the corresponding position of G1 in both the lists is same, for the first element the distance is 0. The next element G2 has been displaced by 1 position and it has been swapped by G4. Hence, the weight of swap is obtained from the distance matrix as pointed and is added to the calculation. Similarly, G3 has also been displaced by 1 position and swapped by G2, as the weight of 0.3 is added to the calculation. For the 4th element G4, it has been displaced by 2 positions. So the displacement of both the positions that come between its displacement need to be considered. Hence, the weight of G4 and G3, which is 0.6, and G4 and G2, which is 0.8, are both added to the calculation. Since the position of G5 remains the same, its contribution to the weight vector is 0. The first equation shows the formula when only considering the pairwise distances, and the second one shows the formula for all types of weights. To evaluate the method, we use the expression of 978 Link's landmark genes for HEPG to cell line, which is perturbed by various drugs and measured at different time points. We took 100 randomly selected drugs and 10 samples for each drug, corresponding to different dosages at different time points. The goal was to predict the correct label of a drug given one of its gene expression signatures. Essentially, it was a 100-class classification task. For gene-level networks, we use reactome pathways and string experimental network. These two diagrams show the pairwise weights distribution obtained from the reactome pathway and string PPI network. In the first set of analysis, we focused on pairwise weights only. To comprehensively compare the performance of various methods, we used leave one out cross-validation, and precision was used as a metric to compare them. As seen in the table, NGSM using reactome pathways or string experimental network performs the best. Further, Spearman correlation and MLP classifier are in the next places. As a point of comparison, note that the precision of a random classifier is only 0.0. The performance of drug precision based on NGSM with reactome pathways is compared to baseline methods. The y-axis shows the average precision using NGSM and x-axis using the Spearman correlation and KNN classifier. Each point in the scatter plot corresponds to the average precision of one or multiple drugs, represented by different colors. Wilcoxon's signed rank test showed a significant improvement using NGSM compared to these methods. Of course, this is a work in progress, and we are hoping to improve the performance by incorporating the other two weights and by evaluating other networks. Thanks a lot for